Look, in the long run, in my life experience, I learned that. So I learned that one agronomist can control disease, or a pest, or in fact, so the human beings cannot control nature. So they try to. Every new technology is linked to the control for something. We, so far, as human society, we were unable to control even a single disease up to now, or a single pest up to now, none. And these people, so they very narrow thinking about one. They think that DNA is the mainly molecule that controls life. I think this argument is totally upside down. I should say that is, in my opinion, we cannot control everything. Why? Because DNA, as I think it was, later on I remember the biology thinker, but the DNA is mainly a dead molecule, because the DNA does nothing. The unique feature of the main features of DNA are, so carries some information, but the DNA itself does nothing about this information, just carry. DNA allow modification, so it's not to DNA control the modification that happens in this, in any base. DNA must be replicated by the cells. The natural selection will control the modification that happens in DNA. So the main issue in biology is the cell. So any thinker or philosopher in biology that take the DNA as a base, I guess they will not survive as a hypothesis or recognition, because this will be not happens in future. So they did the same with when the recombinant DNA technology was developed. People believe that the scientists will have only two limitations: find the sequence that they need to do some first. The imagination, so to do one thing, and find the sequence to develop the new creature that. In fact, 30 years after, we have a few modifications, so the limitation is more wider than the preview in that period. And also with this technology, again, I think DNA is a remarkable molecule that you can do everything with DNA, and its continuity as DNA. Say, in other words, you can boil DNA, the double strand can separate the other, but they can rejoin itself and continue with the same genetic information. And we can build synthetic molecule DNA. So DNA, in terms of biology, is almost a dead molecule in the sense that the function or the make something. One enzyme make one bond between molecules or atoms, but the DNA does nothing. So the cell does everything to replicate the DNA, to translate, to transcription, the information, and do everything else. So, of course, we can, if you can change the sequence, we will provide new traits, new features of the organism. Okay, but this is not the same way that you can conclude that you control life or evolution, because this modification usually have pleiotropic effects. As we did one study, Carol did this study. So the transgenic soybean, the transgene that was inserted in the soybean to make the soybean resistant to herbicide, provides also effects on the metabolic pathway in that affects the relationship between soybeans and microorganisms. Then soybean plants became more susceptible to the disease. So this is a kind of pleiotropic effect. So by making one modification, you do not control life. In fact, it's the other way around. So you make some conditions or alter some other traits that you lose totally the control of the function or the objective of the design or the protocol. So the output will be totally different of your plans. So the another issue that is linked to this one is that these people have this Mendelian view of genetics. For Mendel, looking for several genes that were far apart, control different traits, okay. But usually the genes one affect another. 98% at least of the genes interact each other within a cell. So then one modification, it may be 2% of the genes, okay, you have a different phenotype. But for most of the traits or the characteristic of one organism is affected by multiple genes. So one modification cannot drive the evolution. Of course, you can provide another variation for the evolution to work with. But we're far away from the control evolution, so far.